Thank you for that very hopeful presentation. Our next speaker is Tihamer Toth-Fejel, and he is the senior research engineer, General Dynamics Advanced Information System, and vice chair of the Nanomanufacturing Technical Group of the Society of Manufacturing Engineers. His seminal book in 1996, Legos to the Stars paper, anticipated much of the subsequent work in modular robotics and led to two projects funded by the NASA Institute for Advanced Concepts, self-replicating kinetic cellular automata and nanofactories. He is currently investigating applications for quantum dot nano devices. So we look forward to your talk. So it'll be 20 minutes.
Two hours?
20 minutes.
I've been misled. Thank you. We've all seen the hopeful visions of nanotechnology where you have little robots like this. As an engineer, my first question is, well, how do we get there from here with what we have now? And that's what I'd like to talk about today. Which way do I point? Oh, works great. When I first heard about nanotechnology back in 78? Yeah, 78. I thought Eric Drexler was nuts, especially when he was predicting that we would see this in our lifetime. He took him 5 years to convince me that he was right. And then for many years, he would have these glorious visions that he first described in his nanosystems textbook, but there was no way to get there from here. This past year, the Foresight Institute with Patel and the Society for Manufacturing Engineers sponsored a conference on producing a roadmap that would describe how we get to productive nanosystems. So I'm going to describe what productive nanosystems are, some of my favorite techniques to get favorite approaches for getting to productive nanosystems, some modules that we might use, and then two applications. We're all familiar with the top-down and bottom-up approaches to nanotechnology. The top-down approach can be thought of as a bulldozer stacking tin cans, and it's amazing that we're getting anything done. On the other hand, this NIST picture, which you've seen before, is actually quite an improvement on what Eigler did at IBM. This was done in 5 minutes just by typing at the keyboard. Eigler had to manually move around his individual 35 Xeon atoms, and it took him all day. And if you paid him minimum wage and assumed that all the millions of dollars worth of equipment were free, this Eigler's IBM ad would have cost somewhere around $10 to the 23rd dollars per kilogram. This is not a good business model. So that's why people talk about bottom up. And chemists have been doing bottom up for centuries. But when you really think about it, what you're really doing is you're taking a concrete mixture and throwing things in and expecting to get Rolex watches out. That's not a good business model either, because the more complicated, we obviously can't build Rolex watches like that. On the other hand, chemists have built very complex, this doesn't work. I'm not going to look at it. Have built very complex structures. However, the more complex you make them, maybe this one works. Yeah, the more complex you make them, the more expensive it gets. So we need another model. And fortunately, we have one. We have one from biology. And what we're trying to capture conceptually is a bunch of robots or CNC machines in a factory. And as manufacturing engineers, we know how to do that. We just got to build the tools first. And that's the hard part. So bottom to bottom nanomanufacturing is when you have deterministic nanoscale tools that build nanoscale products and other tools. And that's what productive nanosystems really are. You have atomically precise structures under programmable control that can build atomically precise structures and more tools. Now, this is not self-replicating because you have no guarantee that the components that the system is made out of are going to be included in the output product stream. However, in the entire manufacturing system, just like we have in the United States and around the world today, you have some sort of parts closure. Now, so you can have inputs from, you have inputs from biology, you have inputs from chemistry, and they feed into the system, and different parts of different types of nanosystems can make different parts that others can use. The important thing is that size is not that important. I mean, yes, size is important. We're talking about nanotechnology after all, but it's the atomic precision that's important, and that's one of the reasons biology works the way it does, is because it is atomically precise. As we heard yesterday about the protein folding, or I should say the misfolding of proteins can cause serious problems at a biological level, and it's because it's not atomically precise, even though the proteins themselves are the same size. So the other thing that we want to emphasize is not novel properties, as the National Nanotech Initiative emphasizes, but machines. We want devices and machines so that we can do deterministic mechanosynthesis, and that's what everything here depends on. So the approaches to productive nanosystems are either from the top down or the scanning probe techniques, and then there's the biological or biomimetic methods, which look very promising. Full disclosure, I'm involved in peripherally in a couple of these. With Robert Freitas is working on that, and I think, as you remember, Ralph spoke about that yesterday. Atomically precise manufacturing, that's Xivex's approach, and then we'll get into the others later. I haven't actually made money off of these yet, but next month that will change. Ralph talked about this yesterday. The idea is that you can use precise tips to build perfect diamond structures. And since Ralph already saved me 5 minutes, I might finish in time. The important thing about diamondoid mechanosynthesis is you can build a nanofactory appliance that plugs into a nice friendly plug over here, so it's quite safe. It's not self-replicating. Even if it does make a copy of itself, it's very controlled. You don't have to worry about things going out of control. This is a system. Nanorobots, you can't build nanorobots. You build microrobots out of nanoscale or atomically precise parts. What Xivex is trying, we just won an award from DARPA. I'm a very minor member of this team. The idea is that you use very conservative approaches using equipment that's in many labs today and IC fabs. And what you do is you use patterned silicon atomic layer epitaxy. You use a scanning tip to pull off hydrogen atoms. Then you bring in disilane, and the hydrogen or the silicon atoms attach only where there are dangling bonds, and then you repeat as many times as necessary. Now, there's been some work. Joe Leiden at Urbana has actually succeeded in building structures that, if you look carefully here, are about 15. This graph here represents this dotted line here. You've got about 15 atoms by 6 atoms high, which is pretty impressive. Now, as you can see, there still needs to be work in terms of making the tip sharper and controlling it more precisely, and that's what the 4-year DARPA project is supposed to do. The other thing that we have to do is increase throughput, and that's using multiple tips. But the thing is, even with 55,000 tips, which I think Chad has been able to do at Northwestern, your throughput is not going to be very high. And this is why the biomimetic protein engineering approach is so attractive, because you get 10 to the 23rd examples, not just 55,000. However, it's difficult. You got to solve the protein folding problem. It's very sensitive to small changes in sequence and environment. Now, it is a low-temperature process, but your product is not going to be, speaking as an engineer and comparing biological materials to, say, steel or diamond, it's just not high performance enough. However, it has been successful. This just came out recently. People have actually designed. Now, this took about 60 people 3 years to figure out how to catalyze a particular reaction. First, they had to figure out what kind of enzyme site you need, and then you have to figure out how the amino acids are going to bend, and then you actually had to produce the sequence. This is a difficult problem, and there is an easier way. This was developed by Christian Schaffmeister. He's now at Temple, and basically what you have is you have something like an amino acid. Actually, they're modified amino acids. Only instead of having a single bond that can rotate, you have a double bond. And what happens is this fixes the configuration, the angle and positional configuration between monomers. And so by using solid-state synthesis, you can build, and this has been done, or Schaffmeister has done this, where you actually have any kind of nanostructure you can design just by picking the right bis amino acids in the right sequence. In some ways, it's a great simplification of protein synthesis. And he has this idea. This takes 20 minutes to describe this whole slide, where you actually use a computer, standard computer, to control different plates with different nanostructures on them. That creates more nanostructures, and that's why this is a very productive nanosystem. Okay. Now, this is my favorite, structural DNA. I think this is one of the most promising approaches to productive nanosystems. Here's 50 billion smiley faces, basically done in 2 hours by one person, Paul Roderman. And the important thing is that he didn't need a $2 billion plant to do this, semiconductor plant. He did this by himself in a lab with the equivalent of a kitchen sink. This has been replicated a number of times. The Chinese did it within a year. If you read the article itself, you'll find out that they talk about all the problems they had with, Taiwan Island over here. And yes, that is a problem. On the other hand, Paul Roderman claimed in his paper that this is so simple that a high school student can do it. I was talking to Mark Sims, the president of Nanorex, and I said, you've got a high school student, why don't you go do this? And sure enough, that's exactly what he did. This logo was done by his high school-age daughter, with some help in terms of direction and what she was doing. How it works is fairly simple. You basically have your M13 genome right here, single-stranded DNA, and then you have a bunch of helper strands, 250 of them approximately. And what happens is you have, for example, one of these strands is 16 base pairs long, and 8 of it matches here, and 8 of it matches here, so it brings the 2 of them together. And you do this 250 times, you get from here to here, and eventually you end up with your little smiley face. So it's conceptually a very simple idea, and the software is not that difficult to do either. The other thing is you can make the staples fold themselves so that you can make them stick out, so you actually have, you can actually write. Once you can write, you got lithography. It's not photolithography, so it's not inherently limited to 2 dimensions. And in fact, the first practical application has already come out. These tiles, if you look closely, the two spots here are indexing points, and then depending on what is being detected, and they're specifically looking for RNA sequences, you can identify different RNA sequences by finding out which bars light up. So this is actually a useful application already, and this has only been in production for, or DNA origami has only been around for 2 years. So the big question is, how do you do your software? Paul Roderman gave me his code, and I looked at it, and I'm a software engineer by training, and it was worse than mine. However, what he did is going to change the world. And what Nanorex has done is they've taken his code, and well, actually not his code, they've taken his concepts that are in his code, and they've bulletproofed it. This will be released later this month, and so by the end of this year, you will see high school science fairs with kids doing stuff like this. The biggest limitation is the cost of the DNA samples. But one of the things I'd like to draw your attention to is order DNA. I mean, you punch that button and it spits out your DNA that you just send to your over the web to the oligomer synthesis companies, and you get it in the mail a couple days later. So basically, you heat it and then you mix it up with your M13, and you heat it up to 90 degrees, then you cool it off, and then you're done. You look at it through your AFM. Now the question is, or the problem is the DNA is not a very good structural material, even though it's a great informational molecule. It's not very conductive. It's not very strong. So what do you do with it? Well, you use it for templating. What do you template? Well, there's a whole bunch of atomically precise molecules that synthetic chemists have come up with. I looked at dendrimers. The problem is they're a little too soft and too squishy. Silsesquioxanes, on the other hand, someone was talking about them yesterday. The trademark name is POS. What you can do with this is you have a silica core right here, silicon and oxygen, and then you can put all sorts of different types of organic groups on the corners. So you have a very high flexibility. Not only that, you can grow them. You can grow them in a way similar to dendrimers, only it's much stiffer. And what you can do is, and this has actually been done. This structure has actually been built. This is not. Rick Lane at U of M has been working on this. We're trying to collaborate and find more funding. The nice thing is that the yields are quite high. Not as high as you get from Ford Motor Company, where you actually get cars. Every single one of them works that you buy. That's why biologists look at what chemists do and they go, 50% yield? You know, my cells will die if that's the kind of reaction success I get. So what you want to do is you want to take the Silsesquioxane nanocubes, attach them to your DNA strands, and that's how you get to build your things. Now, that's only half the problem. That will arrange the nanocubes where you want them. However, you need to connect them. And here you need something that is triggerable, preferably something that's photochemical. And there's a number of reactions that need to be looked at in more detail to see what we can do. But if you can do that, then you can build a primitive nanofactory. In this case, with Chris Phoenix and I worked with the NASA Institute for Advanced Concepts. And what we figured out is, well, you should be able to build a water filter. This is a real water filter that really does, you can buy it. However, the actual filter is not very good. It's not atomically precise. We should be able to build atomically precise water filters. Now, what do you do with that? I mean, yeah, you want clear water. There's a high demand for it. There's an even higher demand for dialysis. There's 1.5 million people worldwide that suffer or that need dialysis, and this is it's not a very efficient process. So we thought, okay, a better filter, we'll be able to do something productive with this. Turns out that there are lots of other people looking at this, specifically for dialysis, millipore especially. But as you can see, millipore, this is the stuff that's commercially available right now. These are not atomically precise. So you're going to have different size holes, you're going to have defects. However, people are starting to get down to using lithography. But again, if you look at the atomic level, these white holes, these white areas right here are pores. This is only 15 nanometers thick, and the pores are about 15 nanometers. It's not atomically precise. However, Gail Pergamet has designed a rectangular structure right here using regular organic chemistry. And what these things do is they self-assemble like this. Now, this works. The only problem is that it's a single molecule thick. It's not very strong, so she has to build support structures underneath that. With nanocube pores, I don't know if you can see this very well, but the idea is you just leave voids. Oh, thank you very much. That's great. And you can see that you can either leave single voids, you can leave double voids, and you can do selective filtration. However, the problem is that kidneys are actually quite complicated. They don't just filter by size, they also filter by charge and by shape. So this would have to get much more complicated before it would actually work the way we planned, and you can actually insert it in a human body. You have biocompatibility issues and other things. So this is actually, it turns out to be a much more difficult problem than we originally thought. So but on the other hand, there's a lot of work, and one of the things I thought was most promising is that you actually have am I running out of time already?
I didn't say that.
Somebody's beeping.
What do you have, actually?
I hear beeping sound. Okay. Anyway, the idea is that you use by artificial organs However, even though they've already completed phase 2 trials, they're predicting another 10 years before you'll see these on the commercial market, which is very depressing. So we kind of gave up. I gave up on this thing. How about a simpler problem? Something that productive systems can find as a low-hanging fruit. And Freitas's Respirocytes came to mind. Now, if you have atomically precise diamonoid that you can build, it's a piece of cake. You build something that can withstand 1,000 atmospheres, because that's how much you could pressurize a diamonoid really microvessel. This is 2 nanometer, 1 micron across. And you power it with glucose. And you can do this because you can work with atomic precision very easily, because you have top-down control. On the other hand, and one of the nice things about this, if you just replace 10% of your red blood cells with these Respirocytes, you can hold your breath for 4 hours. So this is better than what we can do with red blood cells, much better. And it seems like a very simple system. You're only working with 2 different types of molecules, and you're either inputting them or outputting depending on exactly where you are, whether you're in the lungs or whether you're in the blood capillaries. And you can tell where you are by just checking the pressure. So we can build pressure transducers now. This looks like it's very promising. Yeah, you have a couple of subsystems. Now, Rob looked at these in great detail in his seminal paper. And the question is, can we do this with DNA organized Silsesquioxanes? And so let's start with logic. First thing you can build is a NAND gate. With NAND gates, once you can build a NAND gate, you can build any electronics. There are some problems you have to deal with. The Bohr exciton radius is about the same size as the nanocube. You also have to worry about 3 dimensions. This is one possible way that you might be able to stack them. But again, you have to connect them, and that takes some new chemistry that we need to figure out. We also you have to do the sensing. You have to know when to release the oxygen, when to pump it in. But we already have people working on this technique. This is a systems problem in terms of putting it all together. Now, one of the interesting things is that because diamond is so strong, we can easily do 1,000 atmospheres. With Silsesquioxanes, we can only get up to 70 atmospheres of pressure because of the tensile strength limitations. But we can make the walls thicker and accept lower performance. On the other hand, a red blood cell, only 1.3 or 0.13 atmospheres are deliverable to the tissues. So we still have a room for improvement. I mean, even this primitive Respirocyte might be able to be helpful in many applications. And what you would have to do is you need to use DNA-assembled shells like this, and you'd have to design them individually using the software that's going to be available this year. Now, the problem is, how do you get these pieces to each individually assemble? And the way to do that, and people have actually done things like this, DNA-programmed nanoparticles, where you arrange them to connect where they're supposed to connect, so that they all become, this is a top and bottom view. So you can build your pressure vessels doing that way. Now, the other thing is you have to pressurize them. You need pumps. These kinds of pumps, okay, with diamonoid mechanosynthesis, are very good nanofactories. You'll be able to do this. But with primitive productive nanosystems, you won't be able to. What you need to do is go with what we have. Now, we have pumps available that people are building using MEMS techniques. And you've got, and once you have a pump, it doesn't have to be selective, but you need a selective filter to screen out either the oxygen or the carbon monoxide, depending on what you're trying to do at that point. And you need actuators. Now, people are working on molecular actuators. You have to be able to use these and the biological systems that we're working with, and people are hybridizing them with mechanical systems. And what you want to be able to do is integrate it with the Silsesquioxane nanocubes. And once you can have that, then you have to put it all together in a system. This is a major engineering product, but it looks like it's reachable. Last slide. I made some predictions a decade ago And I said in 20 years, molecular nanotechnology will cure the common cold, cancer, and make us live for as long as we want, things like that. As we get closer, we see we're pretty much still on track, but the details are getting clearer. I mean, dialysis will get less expensive, it'll get better. The Respirocytes look like they're a low-hanging fruit. We might get there first. People have talked about cancers here, but in terms of the robot that goes out and searches for cancer, that's still a long ways off. And common cold, anything that takes a lot of memory and a lot of manipulation is going to take a lot of work. We have to work with very simple systems first. We need funding, we need advanced engineering tools, and we need clever biologists and engineers, so get to work. Thank you very much.